Antibodies are normally human proteins of the immune system. So it's a protein that every one of us has, and they are able to recognize foreign antigens, so foreign molecules like pollen, for instance, or viruses. Every protein, every antibody has a specificity against one specific antigen. What we do in bispecific antibody technologies is we combine the specificities of two different antibodies into one protein. The bispecific antibodies is able to stick to two different antigens and block them or activate them to do something that they usually should not do. You can use also bispecific antibodies to drive what we call a new biology that normally does not take place. And here I'm thinking about T-cell engager or T-cell redirecting approaches, where with bispecific antibody can grab with one entity to the tumor, and with the other entity it can grab an immune cell and bring tumor and immune cells in close vicinity. And by doing so, the immune cells gets activated and is able to kill the tumor cells. So if you imagine that these two pens are two antibodies, one antibody directed the antigen green and one antibody directed against the antigen red. What we are doing with our technology to build up bispecific antibodies is to combine the red and the green into one molecule like this. And this new molecule is now doing the job of two. We were able to add an additional specificity to the bispecific antibody, creating a trispecific antibody, having the specificity for three different antigens. So we can use the trispecific antibody to capture more specifically and in a more sophisticated way the immune system and attract the immune system to the tumor. I'm very optimistic about the future in science. I hope that we will be able to cure the incurable in the future.